Check 1, 2. Oh, it works. Praise the Lord. It's good to see all you folks, and we really pleased that you came out and hear a wonderful presentation by a wonderful gentleman. Let me say a few words about our speaker this evening, Frank Sherwin. He's with the Institute of Creation Research in Dallas, Texas, and he has given previous presentations with the Creation Weekend celebration here with RMCF back in 2011. So if any of you went to that, you may remember him from that time. And I think we might have had him back one time since then, I think, yeah. Anyway, Frank got his BS degree in biology from Western State College in Grimston, Colorado. And he then attended graduate school, University of North Colorado, studied under the late Gerald D. Schmidt, one of the foremost parasitologists in America. And in 1985, Frank obtained a master's degree in zoology. He published research in peer-reviewed journal of parasitology. He contributes his scientific expertise to a variety of ICR's publications on the creation science issues. For example, Guide to Animals, The Ocean Book, co-author, The Fossil Record, Undermining Nature's History of Life and the Human Body, An Intelligent Design, is one of the ICR's most sought-after speakers. So, accolades for Frank. So I'm going to let him tell you the rest of the story. So you come up and do the rest of it, Frank.
All right. Well, thank you very much, Ed. I appreciate the introduction there. And good evening, everybody. It's always a privilege to be back in Colorado. I'm back home, as far as I'm concerned, you know, having been to school and all that. And seeing Bob and Cindy Borkovic, would you folks wave, please? Bob and I think you met Cindy in Gunnison, right? No, that's it. I knew that. And Bob and I were at Western State College in Gunnison back in and so we had a good time there in Gunnison. And then I want to introduce my second daughter, Maria. Mars, would you wave, please? Maria got her degree at Indiana Wesleyan University in marketing, so we call her Marketing Maria. And she is in La Jolla, California. And she has to suffer, but she's going where the Lord has sent her. And so she's there. It's good to see Ed. It's good to see a lot of folks. A lot of Eds here this evening, too. Well, the average speaking speed is about 190 words per minute, but I'm going to clip right along at 350 with gusts up to 400, okay? And as that infamous King Henry VIII of England told his 6 wives, I won't keep you long. One of those time-release jokes. And I was telling people about that I was going to speak at RMCF, and they go, oh, right, yeah, Rocky Mountain Chocolate Factory. I'm going, no, the people are sweeter here. And so, oh, yeah. And so we're going to talk about insects. My field is parasitology. You don't have to go to France to study parasites. It's another time-release joke. But because, you know, to be a good parasitologist, you have to know your entomology, the study of insects. And so I took entomology, and I enjoyed it very much, and as well as parasitology there at the University of Northern Colorado. I spoon-fed the theory of evolution. I took a 500-level course entitled Evolution at UNC grad school. So I got a real good idea of what the evolutionary naturalists are saying. It's not like I've been raised in some kind of fundamentalist hothouse, you know, Billy Bob's Backwater Bayou Bible College and School of Electric Fan Repair. Yeah. And so, not denigrating anybody tonight, and that's not the purpose of ICR, okay? That's, what we want to do is just kind of look at the evidence here this evening and give glory to the Creator, not the creation. And that's what the Apostle Paul was talking about in Romans chapter 1, wasn't he? And so let's go ahead and get started this evening, and just kind of a kickoff with the ICR Discovery Center. Lord willing, we are going to raise the money to get a museum that will glorify the Creator and not the creation. There's plenty of museums in Texas, particularly North Texas, the Dallas area. What else is there to do in Texas? You know, in Southern California, where Maria got her high school degree, at least they had the mountains, and they had the ocean, they had the desert, and I went to Shadow Mountain Community Church. We were members there, and I would take my son Roy the boy. He's a real joy. And when he was just a little guy, he and I would go on the father-son weekend that Shadow Mountain put out, and we'd spend our time in the desert there. It was just a wonderful time. A lot of what we call laughing and scratching. And that's what we say in Texas. But anyway, so there was a lot of fun there, but there's not too much going on in North Texas. And so what we're going to have, Lord willing, if the money comes in, if you know somebody has got some dinero, let them know that ICR is trying to build a museum. It won't be anywhere as large as the museum in Kentucky, but we're very excited because we are going to have a planetarium. We'll have a number of other items there as well. So this is to keep you informed, the RMCF, of the Discovery Center that ICR is going to be putting in the Dallas area. Check us out, www.icr.org/store if you want to order anything. I didn't bring any books with me. But you can just get the website address there, icr.org/store. Okay, let's talk about insects real quick here this evening. They have 3 pairs of legs and usually 2 pairs of wings borne on the thorax. There's about 750,000 living species, last time I counted. And there's just a whole bunch of different kinds of insects out there. They are truly ubiquitous. We find them all over the planet. And here's a delicious grub. Oh, boy. And so big or small, you know, insects have always been insects. Here's a walking stick. Check that out. This thing is very formidable, and of course it's fairly docile, and it is very well camouflaged, as you see here. Here's a tree lobster from Ball's Pyramid in the Tasmanian Sea. Now, why do they call it a tree lobster? Well, it's a pretty big bug, and they thought they were extinct until they started looking around there in this particular island and found out it's the heaviest flightless stick insect in the world. Wow. But we have not only very large insects, but we have very small insects. The world's smallest free-living insect you see up there seems to have now ended with the precise measurement of the featherwing beetle species. This has recently been retrieved from fungus in Colombia. The smallest individual measures an astounding 0.325 millimeters. Well, the question of the evening is, Frank, big deal. What's 3.325 millimeters? Well, the edge of a dime is 1 millimeter. So this beetle, Coleoptera, is less than a half of the edge of a dime. We're talking really small, real small. And so 0.325 millimeter, but it's a Coleoptera. It's a beetle. Here we have the evolutionary explanation for the origin not only of the arthropods and the insects, but also the other groups of animals, including the sea stars that you see up there and the various other types of creatures. And then here's the phylum Arthropoda right here, and this is what we're talking about tonight. What about it? Charles Darwin's infamous book, published in 1859, was on the origin of the species. One thing he never talked about was the origin of the species. He never got around to addressing that. Why was his book so popular? Darwin's book was so popular in 1859 as it is today to some extent because it explained creation without a creator. It explained creation without a creator. So that's why Darwin's book is so popular. The scientific evidence that he gave back in 1859, of course, none of that scientific evidence fits at all today, but it was popular because of its naturalistic assumption. Well, one quote from two evolutionary naturalists writing in a secular zoology text just a few years ago said, features of the most recent common ancestor for what? All bilateran animals have been much debated. ICR predicts that they will always be debated. They will never come to a full knowledge of how these origins came up in a strictly naturalistic sense, because we read in Genesis chapter 1 that God created what? After their kind. Okay, for you theologians in the audience, how many times do we see that phrase in Genesis chapter 1? After their kind, after his kind, after its kind. It's more than 3, and it's less than 50. The answer is 10 times. 10 times God has told us in one chapter that he created after their kind. So for the Christian, that should settle it once and for all. God did not use the slow and gradual evolutionary process, because it says in the book of Beginnings, the very first chapter, that he created after their kind. So we shouldn't find the kinds of connections that the evolutionists are looking for. Again, we're not criticizing them, but we just think it's an exercise in futility. So this was said way after Darwin's book in 1859. This was said in the 21st century, has been much debated, the recent common ancestor. Here's a bilatera here, and supposedly whatever this common ancestor was, and by the way, it's never been found, and we predict it will never be found, gave rise to these major groups here. Now, these solid blue lines are deceptive. The solid blue lines you see up there are deceptive, because that indicates direct evidence, direct evolutionary evidence, fossiliferous evidence, whatever you want to say, but it's direct evidence. So in order to be more reasonable or honest, if I could use that word lightly, what should those lines be? They should be dotted. They should be dotted lines. Back when I was in college, they had dotted lines that inferred an evolutionary relationship, not a solid line that dictates that that is actually what they found. So when it comes to the origin of the phylum Arthropoda, this comes from the University of California website. They are honest in admitting that they don't know what this common ancestor is, and I salute them for that. But from this unknown common ancestor, and they put the question marks here, gave rise to the various groups that you see here. But they simply don't know what that common ancestor for the arthropods are. As a matter of fact, way back in the year 2015, a secular author said, quote, "The question here is, how do we go from something that looks nothing like an arthropod to something that looks completely like an arthropod?" End quote. That's a very good question. I salute him for his intellectual honesty in saying this is the big question. We will say that's always going to be the big question. They will never find that common ancestor. Again, just last year, an arthropod, which lived on the ocean floor for about 500 million years, would have looked very much like today's shrimp, say paleontologists. 500 million years of no change. What is a one-word definition of evolution? Change, okay? So for 500 million years, what we have is that shrimp have always been shrimp. Delicious, chilled shrimp. Well, what about this tree of life? Great disagreement exists, however, about how the animal phyla are related to one another. Again, this comes from a secular zoology text by two secular authors published just 3 short years ago. Great disagreement about how these animal phyla are related to one another. And again, you see on the screen, they have these deceptive solid lines, indicating that there's direct evidence when in fact there is no direct evidence. They are deceptive. Recent studies have cast doubt on the historical viewpoint that sponges are sister to all the animal lineages. Here's the porifera, or the sponges right here. And again, I appreciate Dr. Whelan and others admitting that this historical viewpoint that the porifera, or the sponges, are sister to all other animal lineages is now doubted. Arthropod fossils and molecules have necessitated major revisions in our understanding of insect evolution, according to these same two secular authors in a zoology text just 3 short years ago. Necessitated major revisions in our understanding of insect evolution. Recent research is causing zoologists to rethink their perceptions of insect evolution. Nothing wrong with that. That's the way science operates. It's self-correcting. I understand that. But we keep seeing this phrase, ladies and gentlemen, over and over and over again. So this idea of rethinking the perceptions of insect evolution is a common theme. This, of course, is a cladogram here, cladistics, that is used. Well, I won't get into that right now, but here's retired zoologist Donald Williamson in England. He said, nobody knows where caterpillars came from. It looks like caterpillars have always been caterpillars. Very profound thing to say this evening. Caterpillars have always been caterpillars. In one beautiful specimen, the 50-million-year-old leaf had been partially eaten by prehistoric 100% caterpillars, just as modern caterpillars feed, and their feeding tubes are preserved on the leaf. So again, it doesn't seem to be how far that you go back in time into the distant evolutionary past, supposedly. Caterpillars have always been caterpillars, insects have always been insects. In fact, when you go to the fossil record, you find that the fossil insects show no evolution. Insects have always been insects. We have fine, clear imprints of insects, particularly the coleoptera, which are very well designed, if I can use that word, to be fossilized and to be found as 100% fossils of the coleoptera and the hemiptera, the homoptera. All of these types. The Lepidoptera are not found, the butterflies and moths, in the fossil record, but they are found, for example, in resin and the various sticky substances that are exuded. And they are, in fact, just as what we have always seen. 4 ancient ectobius species were recently discovered, well, where? Right here in Rifle, Rifle, Colorado. And again, it's supposedly 49 million years old, but it's still the same genus that we see today. There's been no change. And more recently, this is in Current Biology just a few months ago, February of this year, the oldest worker and soldier termites were 17 million years old up until February of 16. Now, 5 termite species are reported in 100 million year old amber from Myanmar. And so when the conditions are right for preserving these insects, like in resin or in amber, we can identify them right down to the genus level and sometimes right down to the species. What's going on here, ladies and gentlemen? Where is the change? Where are the evolutionary ancestors? Insects have always been insects in the various groups, essentially, that we see today. The oldest beetle fossils. Beetles, again, are ubiquitous. We just find them all over the place. One evolutionist said back in, I think in the 50s or 60s, the creator must have an inordinate affection for beetles because he made so many. Well, that's true. Beetles are a very, very important part of the ecosystem. We don't make any apologies for that. The fact is, we have to ask the question, where did beetles come from? What was their origin? From the lower Permian are approximately 300 million years old. A 296 million year old insect has been reclassified as a beetle, pushing back the origin of beetles by millions of years. And so the question we ask this evening is, where is the evidence for beetle evolution? An international team of evolutionists made a surprising find that a 20 million year old fossil they discovered belongs to a species of aquatic beetles which is still alive today and widely distributed in Eurasia. Again, this was just last year. So for 20 million years, it's just the same kind of beetle. There's been no change. Beetles have always been beetles, yeah? This is amazing. This is absolutely incredible, and I think most of you know about this. I mean, this is right up there with the soft dinosaur tissue. We've all heard of soft dinosaur tissue, the fact that we're finding it all over the place now. Insects' bloated abdomen carries traces of blood molecules that are 46 million years old. There's also oil and plant heme pigments within the formation found in Montana. But to find organic material, allegedly 46 million years old, is it's quite a stretch. And so this is very, very interesting. We believe by faith the Genesis flood occurred approximately 4,700 years ago. So to find organic material ensconced in some of these fossils, to find soft dinosaur tissue is not surprising at all. But to say that there's 65 million year old soft dinosaur tissue, and then Mary Schweitzer wrote a second paper with 13 other evolutionists. This time, the sample was from a different dinosaur, dated at 80 million years old. She was very good in doing a double-blind evaluation. And again, soft dinosaur tissue dated at 80 million years old. Well, few events in the animal kingdom have been subject to as many hypotheses, educated guesses, as there's that key word again, folks, the what? Origin of insect wings. Well, Frank, look at that date. That is 1978. That's a while back when that was said. But said it, they did, that it is a subject of many guesses, hypotheses regarding the origin of insect wings. Well, let's look at this very common zoology text by 6 evolutionists. As a matter of fact, when I went to Western State, we used Hickman, Hickman, and Hickman in our zoology class. They said in 2011, in the 21st century, the evolutionary, there's that word again, origin of insect wings has long been a puzzle. I appreciate that they said that, since other evolutionists have been saying that since at least 1978, and of course way before that. A whole body fossil impression of a flying insect discovered in the US dates back to 300 million years. That was said back in 2008. And going to the ever-reliable Wikipedia, Wikipedia said the evolution of insect wings has been a subject of debate. And Wikipedia is certainly not a friend of the Bible-believing creationist, but they're intellectually honest in admitting that they don't know where insect wings came from. The first time you find winged insects, what are they, class? They're winged insects, right? Romans 1:20 says, the Apostle Paul says that God's creation is not only seen, but Paul takes an extra step and says God's creation is clearly seen. Clearly seen. Sherwin translation is painfully obvious. God's creation is clearly seen. Why? I think it has a lot to do with God's love, don't you? God loves us so much, he wants us to see and understand his clearly seen creation. Doesn't Paul say that in Corinthians? That God is not the author of confusion. No, God doesn't want to confuse us. He wants us to know. He wants us to understand. And therefore, he has made his creation, even though it's been corrupted by sin, Paul says it's clearly seen. So yes, God did curse the earth, didn't he? He cursed the earth with thorns, right? And what else? Yeah, thistles, thorns. I think probably parasites too, although I can't be dogmatic. Why? It doesn't say that. Yeah, it doesn't say it in Scripture, but I think the parasites would fit right there with the thorns and the thistles, parasites, definitely telemarketers. And so... But even though we do have this curse on the creation, we understand that God's creation is clearly seen. So living things like insects clearly looked like they were designed. So how do evolutionists know they weren't designed? What is preventing us, ladies and gentlemen, from going down the design inference? I mean, if that's what it looks like, there should be no reason not to pursue that, to investigate that. Certainly, design means what? A designer. Creation means a creator. If this solar system looks tailor-made, there might be a tailor. Psalm 111 says, "The works of the Lord are great, sought out among all of them that have pleasure therein." His work is honorable and glorious. He hath made his wonderful works to be remembered. Boy, I love that verse. Those series of verses there in verses 2 through 4 are simply wonderful from a creation science perspective as we look at God's living creation. Morpho butterfly. Boy, do I love those things. I've been down to South America twice now, and when I was down there both times, I got morpho butterflies, the big ones, Lebamba, the big morpho butterflies in these marketplaces and all that are designed. They're found inside, they put them inside of a glass case surrounded by wood. It's very ornate, very beautiful. And so I took a lot of care to bring them back to the States. And I would show them in churches. I'd show them various creation meetings until finally I managed to break both of them. But it was fun while it lasted. So if you notice with the iridescent shine, the sheen, the blue kind of a turquoise color that you have there, that's very, very unique. As it turns out, it's not due to any pigment cells. There are no pigment cells in the top of the wings of these morpho butterflies. And by the way, these same colors are found in other invertebrates, such as what we find in tidal pools, a number of other places where we're beginning to find these, what they call biophotonic structures. But the kind of structures that you see in the butterfly wings there are also found in blue jays. It turns out that blue jay wings do not have any kind of pigmentation cell. They also have the kind of structures that we see here. What we're talking about are structures that are very, very small, that are found in the scales of the morpho butterfly. It comes from a complex, what we call nanostructure in the wing scales. So what you have to use in order to see it is an electron microscope that magnifies things. We all know what an electron microscope is. It's what you did not use in high school and college. And so using a scanning electron microscope, you take just one of the scales and magnify it thousands of times, and this is what you find. These structures, which are so sophisticated, so detailed, each one of these little ridges here are measured at 0.00004 millimeters. So each one of those detents there, 0.00004 millimeters. And what that means is that white light, which contains what? All the colors of the rainbow. This is, was it Physics 101? White light has all the colors of the rainbow. When the white light strikes these structures here, they're called nanostructures, or I've read in some literature called gyroids. Most of the wavelengths are absorbed effectively here, but one wavelength of light is reflected back to our eyes. Guess what wavelength that is? Blue. See how easy science is? It's that turquoise blue color that is reflected back. All the other wavelengths of light are absorbed, and that's the nature of light, isn't it? When it strikes a solid surface, it's either absorbed by that surface, or it goes straight through, or it's reflected back. And so that's kind of very, very interesting that we see there. This is very, very specific, cut at a very, very specific sequence, each one of those. Folks, I don't attribute this to blind, dumb, lucky mutations, mistakes, right? Beneficial mutations. It's kind of a, what do they call it in English? An oxymoron, right? It's kind of like saying jumbo shrimp. Is that oxymoron? My favorite is rap music. Microsoft works. Okay, so there might be a beneficial mutation, and I know one, and that's red hair when I was growing up. Definitely a beneficial mutation. But other ones I'm kind of questioning. Anyway, it says 3 designs that give wide-angle reflection, a Christmas tree-like shape that we've just seen there, alternative laminae layers, and a zigzag pattern of the ridges, each at 0.00004 millimeters. So this results in the reflection of up to 80% of the incident blue light. So all the colors of the rainbow strike these specific nanostructures you see here, and it reflects back just that blue turquoise light. Just fascinating, isn't it? Wow. A countercurrent heat exchanger for you engineers out there, okay? We are finding now countercurrent heat exchangers in the arthropods, the insects specifically, or the Lepidoptera as we see here. Helps maintain a high temperature in the thorax where the flight muscles are located, allowing the endothermic insect to fly in winter. That almost sounds like an oxymoron. Endothermic insect? We usually attribute endothermy to the vertebrates, certainly not the invertebrates, but this is just a fascinating, I think, design inference, don't you? They need to have their muscles warmed up so they can fly, and so the Creator has designed it in such a way that you see there, the flight muscles are located. I love this. Look at that. This is a guy right here, a male moth. Most information a moth receives comes from its highly developed antenna, which provides it with an incredibly sensitive sense of smell. The antenna of male moths is so sensitive. How sensitive is it? That in some species, they can detect a single molecule of a female moth's sex pheromone in a cubic yard of air, allowing the male to detect her from 8 miles away. One molecule. In other words, the male is flying along, as you see here. The female, 8 miles away, releases a sex pheromone. It gets significantly diluted in its trip towards Mr. Moth, right? Until there's just one molecule, one organic molecule. It strikes this antenna array that you see here, and it rings a bell. And we know it's successful. Why? We still have these moths. Okay. Here's a compound eye of a fruit fly. You know, nothing simple about that. The compound eyes of these arthropods are simply amazing, especially my favorite arthropod, which is the trilobite. We don't have time to get into that this evening. Paul Gabrielson said just 3 years ago, an insect's compound eye is an engineering marvel. High resolution, wide field of view, and incredible sensitivity to motion, all in a compact package. So this is just absolutely amazing to look at the light structure and to see how it all interacts. Well, here's a question in the evening, folks. Do we normally attribute these high-precision gears that you see in the picture up there from an explosion in the steel mill? Shake your head like this. No, you don't, okay? And we find that these gears are very sophisticated, very detailed. Nothing is left to chance when you build gears like that, right? Whether it's gears that you find in a car, you know, transmission, or in the old-time pocket watch. Remember your great-grandpappy or your pretty good grandpappy used to have? Okay, and so gears require an engineer, requires an architect. Well, let's look at a scanning electron microscope as we take a picture of this critter right here, a plant hopper nymph. These are the legs of the plant hopper nymph. It's a very, very small creature, and a British evolutionary naturalist decided to take pictures using the electron microscope of these plant hopper nymph. And so he got out the equipment, he got a volunteer plant hopper, and he took a picture. And he said 3 years ago, quote, "What we have is a prototype for incredibly small, high-speed," what's the next section there? High-precision gears. High-precision gears. A guy who said that is Dr. Gary Sutton at University of Bristol in England, and said it in National Public Radio. Here's the picture he was talking about that he took with the electron micrograph. This is an evolutionary publication, and so they're the ones that use the G word here, gears, showing how that they interact, as you see here, so that the plant hopper, when it hops from there to here, does so with an extreme fidelity. He doesn't have to kind of guess. The way the gears are constructed, the plant hopper knows that if he aims in this direction, that's where he's going to go. So again, I would submit to you this evening that nothing is left to chance when something like this was, could I use the D word, designed? Well, Dr. Sutton, as an evolutionary naturalist, would not attribute what you just saw to a creator. As a matter of fact, he knew people would think, boy, sure looks like it was created. And he says, no, it wasn't. And this is what he said in his publication. These gears are not designed. I thought that was that's pretty graphic. And it's, you know, I don't know, I could say a lot of things, but he's saying these gears are not designed, they are evolved. representing high-speed and precision machinery, evolved, there's a word again, for synchronization in the animal world. And so I think he's basically willingly ignorant. He's just not going to entertain the idea that those gears, which look so obviously designed, were designed. That reminds me of a verse in the Bible. It's found in Romans chapter 1 and verse 20, where Paul said God's creation is clearly seen, okay, clearly seen. And so what we would maintain, ICR, we kind of looked at Dr. Sutton's quote, as you see here, these gears are not designed, they're evolved, representing high-speed precision machinery. And so we did a little bit of a variation on that theme, and we said these gears are created, representing high-speed and precision machinery designed for synchronization in the animal world. Turnaround's fair play, okay? We're going to give glory to the Creator and not the creation, okay? So what we have done is to use creation scissors and cut away that evolutionary blindfold where they refuse to see the evidence for creation. So use creation scissors, put on your creation glasses and take a look at the world around you. This reminds me so very much of the late Francis Crick, who was an evolutionary atheist. He's not anymore, but Dr. Crick said in his 1988 book, look what he said, biologists must constantly keep in mind what they see was not designed, but rather evolved, okay? Remember what Dr. Sutton said a few slides ago? He said these gears are what? Not designed. He was so concerned that people would come away looking at his electron micrographs and coming to the conclusion that design means a designer. So he said in a paragraph that these are not designed. And here's his fellow Englishman, Francis Crick, co-discoverer of the spiral helix DNA molecule, said, we have to keep in mind that what we see was not designed, but rather evolved. In other words, God has no play. God has no say in what we see in the world around us, no matter how sophisticated, no matter how detailed. Well, I have this genetic text in my office. I need to get the update. I'm sure they've probably updated it since 2012. But one thing that the university-level genetics textbooks that I go through on a regular basis at ICR is looking for that section of the textbook on beneficial mutation. And if there's no section, maybe there's a chapter. And if there's no chapter, maybe they have a sub-portion of the chapter, or maybe a table, a diagram, a chart, something that would list for the university student all the beneficial mutations that we would expect based on evolutionary naturalism. What we have here is the Antipedia mutation that has the antenna of the Drosophila, the fruit fly, and under certain conditions of mutations, it will turn those antennae into legs. It's called the Antipedia mutation, and kind of ruins a fruit fly's day. You have legs coming out of your forehead. It's a mutation, yes, but I would question whether it's a beneficial mutation. I had a debate with an evolutionist at Denver University back in the mid-1980s, and he was member of the Department of Biology there at Denver University, and he showed this picture during the debate, a four-winged fruit fly, where the halteres are the balancing organs of the Drosophila, are mutated in such a way that they become wings instead of their balancing organs. And sure enough, you have a functional pair of wings. The bad news is the fruit fly doesn't really fly very well. As a matter of fact, it would make a nice hot lunch for any predator because it flies kind of in circles. It's very, very uneven flying patterns and all that. So again, we would openly question whether having an extra set of wings would be what we would call a beneficial mutation. The reason why I have Drosophila up here is this is the most common animal used in genetics. They're very small. They have a high turnover rate. They only have a couple of chromosomes to work with, and so you can start to be very specific as to what mutations can be found on what chromosome. But a very ambitious project was started to justify evolution using natural selection back in the mid-1970s. And they went through 600 generations of Drosophila, 600 generations. They finished in about mid-2000, and what did they have at the end? Drosophila, fruit flies. So we could say fruit flies have always been fruit flies. It's a fascinating study. It's a very long paper, and it's published in Nature magazine. And I can give you the reference for it, but I read it, and it was very, very interesting. So from beginning to end, they've always been fruit flies through the 600 generations. This is Ridley's third edition of Evolution. 2 pages out of 751 pages addresses beneficial mutations, but no specific examples of beneficial mutations are given in this very formidable book. Kind of interesting. By the way, Steve Gould, in his book Structure of Evolutionary Theory, which is well over 1,000 pages in length, the title of the book is Structure of Evolutionary Theory, but he never got around to discussing beneficial mutations. And he knew he was dying. He knew he wanted to leave his mark there. Well, let's talk about God's amazing dragonflies. I love dragonflies. They've always been dragonflies, yeah. Fossil dragonflies have been found with almost a 3-foot wingspan, well, 2.5-foot wingspan there. Here's a fossil dragonfly. And again, this is measured in millions and millions of years, but it's still a dragonfly. It's neat that we can find these because they're so delicate, but when we do find them, they're always dragonflies. They're expert flyers. They can fly straight down and up or hover like a helicopter. If they can't fly, they'll starve because they only eat prey that they can catch while flying. Some adult dragonflies live for only a few weeks. Others live up to a year. Their eyes have up to 30,000. Look at that eye there. It's almost the entire head is all eye. 30,000 individual lenses. Fruit flies that I just talked about have about 750 individual ommatidia. So this is why it's so difficult when I was in graduate school trying to get dragonflies, because they can see you coming a mile away. I mean, their whole head is just eyes there. And you take one of those dragonfly nets or insect nets, and you start your swing. When you see the dragonfly coming about a half block away, you have to start your swing and hope that they'll, and right at the last minute, they always go, every single time. Well, some entomologists, zoologists got frustrated. Guess what they started using very successfully? Shotgun. Now, they didn't use a really heavy shot. They used a very fine pepper-like shot, almost like talcum powder, which would just kind of mess up their wings a little bit and ruin their day. So they would use shotguns, and it was very effective in getting them. But I love today's fun fact, okay? And this is the ommatidia right here. It's a very, very sophisticated unit of vision here. It's got 2 different lenses, pigment cells, receptor cells. There's a lot more to it than this. I used to have my zoology students draw this cross-section. Dragonflies have about 360-degree vision. They keep mosquitoes at bay, and they're indicators of clean water. So when you see dragonflies around a waterway, the water there has got to be pretty clean. U.S. Fish and Wildlife Service said you might call dragonflies the stunt pilots of the insect world. They wear flashy colors, they dart at speeds up to 30 miles an hour, boast ancestors that predate dinosaurs, according to the U.S. Fish and Wildlife Service. But again, they're still 100% dragonflies. Very good. A dragonfly, look at this, I just found this recently. A dragonfly barely 1.5 inches long appears to be animal world's most prolific long-distance traveler, flying thousands of miles over oceans as it migrates from continent to continent, according to newly published research. A dragonfly. Think about the energy reserves it has to have. It can't have too many, otherwise you'd kind of run out of steam, it'd be too heavy to fly. But it can't have too little because it would starve to death. It has to be just right to get to where it's going. But the reason why I like dragonflies so much is because of the dragonfly larva. These things are savage. They're wicked, whatever other adjectives that you can use. But these dragonfly larvae really, really are the tigers of the pond. And you ask any fisherman who digs around in a freshwater pond or waterway to get some earthworms or whatever else, they'll get nailed by these things. And yaha, it makes you comment. Here is the anterior portion of the dragonfly, odonate mouth part of the dragonfly larva. It's got the labrum, and the labium comes out. It's called a mask, and look at these two wicked pincers coming out here. These things can shoot out, and by the way, you can go to YouTube and type in dragonfly larva, and you can see these things in action. They're wicked. This is 1999, the predator larva. This is the same larva that you see here. It's grabbing a vertebrate. So here's an example of an invertebrate feeding on a vertebrate, such as a minnow, or maybe even a tadpole larva, or small frogs as well, if they're hungry enough. And then here's an actual picture of a dragonfly larva, and it's actually found and is feeding from a minnow that is captured. So the dragonfly nymph looks like this, and it likes to get, as I say, something like what is that? A tadpole or a small frog or something like that, a vertebrate. So they, again, these very wicked pincers here will do a good job there. And here's dragonfly larva with its labium extended. And so one photographer wanted to get a good picture of the dragonfly larva, and so he used his camera, got up real close to the dragonfly larva that his friend was actually holding in his fingers. In Texas, we call these fingers, fingers. And so the photographer got real close, and here it is. And this dragonfly larva is alive, and he is mad. It doesn't take much to get a dragonfly larva mad. And you can see the wicked pincers here, and as he was taking the picture. Well, that brings us to the giant sea scorpion. I had to say something about that because Simon Brady at the University of Bristol in England said, "We have known for some time that the fossil record yields monster millipedes, super-sized scorpions, colossal cockroaches, and jumbo dragonflies that we looked at. But we never realized until now just how big some of these ancient creepy crawlies were." And so this is the giant sea scorpion here, and I'm kind of glad those things are extinct, okay? I was openly skeptical that these things got that big, and so I did a little bit of research, and the New York State conservationist just 3 years ago said, yeah, some species grew to 9 foot long, the largest arthropods ever, 9 foot. Boy, you imagine the damage they can do if they got hungry. By the way, something I didn't mention a few minutes ago when I was talking about the dragonfly larva, remember what we believe to be the pre-flood world, these dragonfly larvae got to have a wingspan of, the dragonflies, of about 2.5 feet, right? Well, that means that the actual larva must have been huge. The larvae had to be big in order to metamorphose into the adult dragonfly with a wingspan of almost 3 feet. Can you imagine wading in water in the pre-flood world and getting your leg attacked by the labrum as it shoots out there? Boy, Well, anyway, have you seen those Florida mosquitoes? Maria was born in Florida, and her 2 siblings were as well. Have you seen those? They're very, very wicked. So again, I'm glad that we're here in Colorado and not in Florida. Psalm 19 says, "Yea, much fine gold, sweeter also than honey and the honeycomb." And bees are just amazing here. What a brave little girl. I love to study bees because they are designed to see in certain sections of the electromagnetic spectrum that we cannot see in, such as the ultraviolet. Here's the infrared here, here's the ultraviolet here, and then this is the visible spectrum that we are allowed to see, and of course we can't see anything around from this area here. Just a very narrow band of light that gives us the visible spectrum. But bees, as I say, are designed by the Creator to get and look into the ultraviolet region. And we've all seen this in junior high, haven't we? In grade school and junior high, where we see flowers as we see them, and then flowers as bees see them in the ultraviolet range here. And so this is how a bee sees this, and then a trumpet flower here, okay? And so we find that, again, they look quite different in the ultraviolet light. which is what, as far as we can tell, how bees would see this, dandelion and so forth. So we see this, and we are blessed by the beautiful aesthetic quality here, but bees are more interested in what we call the nectar guides. Nectar guides are visible to bees, but not to humans. Here we have the beautiful flower here, and how the bee sees it with their incredibly designed eye that we see here, compound eye. And so the ultraviolet image generates 3 rings of color to guide the bees. Nectar guides or floral guides are patterns seen in some species of flowers, guiding pollinators to their rewards. Rewards commonly take the form of nectar, pollen, or both. And this is the pigment which plays the role of the nectar guide here. There's nothing simple about this organic molecule. Very, very sophisticated, playing the role of the nectar guide. Well, here's a typical Ommatidia, and this is a lot more sophisticated than the one I showed you a few minutes ago. The brain of a bee is composed of a mere 950,000 neurons. That sounds like a lot, but it really isn't. 950,000 neurons, or nerve cells. That's about 0.01% of the neurons found in the 3-pound human brain. 3-pound human brain, by the way, is the most sophisticated accumulation of matter in the known universe. And so this is just 0.01%. Using this tiny bee brain and associated vision, bees have been able to solve complicated color puzzles and even recognize human faces. They do this by using their 6,300 Ommatidia that comprise the eye. Bees also have been created with the ability to distinguish up to 300 separate flashes of light per second, an attribute they use as they rapidly fly over the changing landscape. And so imagine that, 300 separate flashes of light per second. That's some pretty good evaluation that they use. So how big is a bee brain? Well, it's about the size of a single grass seed. So you have one grass seed, and that's approximately what a bee brain is, 950,000 neurons in the honeybee brain. Here is a honeybee brain again, using a CT scan of the bee brain. Foraging insects like bees have learned to deal with a complex array of sensory, landscape, and chemical cues while they search for pollen. So we see some very, very sophisticated detail here. Researchers at the Vision Center have demonstrated that even on days when the sun doesn't shine, bees can navigate to and from a honey source by reading the pattern of polarized light in the sky, and then explain to other bees where to find it with their famous waggle dance. We've all learned about that, right? In junior high, grade school and all, how the bees wiggle around in the hive there to tell their friends where to find it. Here is the bee vision going into the ultraviolet here, and the human vision stops right at around 400. And so it's very, very sophisticated. A series of clever experiments by the French scientist 3 years ago has shown that honeybees have a skill thought unique to primates. Conceptual relationships. When presented with varying patterns in mazes with rewards, they appear to understand left and right, up and down, and same different concepts. So bees seem to be pretty bright. You can't come to any firm conclusion about this because you can't interview the bee, but you can come to some pretty interesting conclusions about what bees are able to do, and they really do seem to be quite bright for having a brain only the size of a grass seed. One researcher at the Australian National University in Canberra has described wasps as wonderful, autonomous, flexible, and robust. He explained that it took more than a decade for researchers to understand how wasps can know places and how they can even shuttle back and forth between them. And this was reported just a few months ago. And then one researcher said, in relation to this article, we want to understand what trick the insects are using to acquire the competence of homing, even, as I say, on cloudy days and all. And just recently, February of this year, the eyes of moths create sheets of graphene that biologists claim is the most light-absorbent material ever made. Quote, we realize that the moth's eye works in a particular way that traps electromagnetic waves very efficiently, end quote. Well, I could go on and on. I've got, I think, 400 more slides in these, but I think you can get an appreciation for just the sophistication that we're finding in the insect world. And these are the invertebrates. These are just the insects. And so as time goes on, and you saw a lot of these references were within just the last 3 years. You can imagine what it's going to be like for the next 3 years to see just the detail, sophistication, and complexity of these fascinating, fascinating creatures. By the way, you've heard about the problems they have with honeybees and the fact that they're, dying off so rapidly. It seems now that they found it has to do with a pesticide. Some of these pesticides that had gotten into their food chain and all. So hopefully we can turn that thing around and say that, because without the bees, you're not going to have a lot of the cash crops, the food crops that we need as people and then also as animals as well. So I didn't know that if there's going to be any time for Q&A, but would there? Okay. Yes, sir, we got microphones right here. And maybe if we want to do that. Good.
Why are there so many bugs and so many types of bugs? Why are they so numerous within each type?
Okay, why there's so many bugs and why there's so many at each type? Well, I don't know. They are a basic foundation of what we call the ecosystem, like beetles, for example, and they have a lot of work to do. I can think of, for example, the dung beetle, which needs to do a lot of recirculating of this material, and what better way than to use beetles to do that on a very large scale? And by the way, ivermectin is so potent that the dung beetle won't even touch it, the dung there, because the ivermectin has a staying power right into the dung there, and the beetles simply won't touch it because it's such a potent pesticide, But be that as it may, I think the reason why we find so many large numbers of these insects is because they have a foundation of the ecosystem. And they got a lot of work to do, for example, as pollinators and all that. And we understand some birds and bats are pollinators, but insects take the cake for that, when it comes to pollinating on a very large scale. So it's a good question. Does anybody have any others? I know there's somebody from University of Colorado here, might be able to. But yeah, it's just a foundation of the ecosystem. Yes, sir. Terry's a good man. Go ahead.
Why did God make the mosquito?
Oh boy, yes. Why did God make the mosquito? That's a good question. Do we have any other questions? Oh, no.
I was seriously wondering, Frank, if that mosquito you showed with the blood in it,
Yeah.
you actually find blood cells in it?
I'm not sure if they've actually found, intact erythrocytes on there. Erythrocytes are about 33% hemoglobin, and I don't think that they would be in any shape. If they did find erythrocytes, that's all they find inside are hemoglobin. We would have to then look for the white blood cells, right? White blood cells have the DNA that are nucleated. And is that what you're getting at, or?
Well, like with the dinosaur blood
Yeah.
cells they've actually found, I was just wondering if they found anything like that.
Yeah, that's the jury's still out on that. But to find these insects, this one insect that had a belly full of blood, it's congealed and all that, but it still has its organic properties. That's very significant. And so, I think jury's still out. They'll still do some work on that. This man here, and then.
It's actually our position, just going back to the mosquito,
Mhm.
in the context of a mosquito needing blood, which is supposedly post-fall or post-flood.
Yeah,
Prior to that time, everything was vegetarian, so
correct.
they deal with carnivores, right? And some carnivores, obviously, that's, somebody's opinion of it. But in the context of mosquitoes, what are they going to live on prior to the flood?
Good question. I did some research on that. Not that my research is definitive by any stretch of the imagination. First of all, we have to keep in mind regarding the creation and then the fall. Dr. Henry Morris, a very godly individual, a very sharp scientific mind, came to the conclusion that between the creation of Adam and Eve and the fall of Adam and Eve, it'd just be measured in days. Very good, just days. Now, there's two reasons for that. First reason is when God drove Adam and Eve out of the garden, did it say Adam, Eve, and all their kids? It just said Adam and Eve. So they didn't have time to what? To procreate. So that's indication number one. Not proof, but an indication. Now, indication number two is the nature of Satan himself. Satan is the deceiver. Satan hates what God has created. Can we all agree on that? Satan hates what God has created. He wants to corrupt as much as he can. So what would cause Satan to sit around and wait, centuries or months or whatever else? He is going to want to corrupt what God has created. And so I believe with Henry Morris that the corruption of the creation is measured in just days. Now, for those days of the mosquito, remember it's just the female mosquito that has the mouth parts used to slice and dice into skin. Right? Okay, and why is it only the female mosquito has those mouth parts? It's to get underneath the epidermis and the dermis and get into those capillaries where the blood is. She needs the blood to make the eggs. Eggs are 100% protein. Males don't. The male mosquito is mellow. He goes to the flowers. Seriously, male mosquitoes go to the flowers, gets the nectar. The female goes for the jugular. But anyway, okay, so the female mosquito has the mouth parts in order to dice and to slice into the skin, down to the capillary to get the blood. So the question in the evening is there something in the plant world that can take the place of hemoglobin, which is what the blood is? I mentioned earlier, 33% dry weight of a typical erythrocyte is hemoglobin, the molecule designed to carry oxygen. The answer is there is. In some plants, there is a molecule called leg hemoglobin. And it's almost like hemoglobin that we find. And by the way, hemoglobin is found throughout the animal kingdom. There's even some bacteria that have hemoglobin. Some worms, annelids, have hemoglobin. Vertebrates, of course, have hemoglobin. But the leg hemoglobin, which is found in some of the nitrogen-fixing plants, is a true proteinaceous compound that maybe the female mosquito could feed off of. How's it going to get in that plant? Well, just the same way she uses as these mosquitoes in the 21st century to slice and dice into our skin. She has the construct of those mouth parts. And when I taught zoology, specifically parasitology, I had microscopic images of both male and female mouth parts. And that was one of the lab questions, is which is male, which is female type of thing. 50-50 chance. But the female has the mouth parts to slice and dice into that portion of the nitrogen-fixing plant to get the leg hemoglobin. So at least there's an answer there. It may not be the right answer, but I think we might be onto something. By the way, what we're called at ICR is the Institute for Creation Research, not the Institute for Creation Answers. Okay, so we're still doing the research, okay? You can't fault us. We're trying to do the research, but we're not genuflecting before Darwin's altar. Question? First of all, did I answer your question, sir? Okay, good. Yes, sir. Yeah, survival of the insects in the flood says 2 of every kind of animal. That would include the insects. One thing that we know for sure in regard to the various kinds of creatures that God created is there's a lot of genetic plasticity, is there not? A lot of plasticity. Some fellow creationists who don't agree with ICR say the biggest evolutionist in the country is ICR scientists, because we believe there's a lot of evolution going on since the flood 4,700 years ago when the ark landed in Mount Ararat, released those animals, and we've seen such diversification since then. Yes, there is a lot of diversification, but it's within the created kind, such as the dogs, right? We have all sorts of dogs, dingoes, coyotes, all these varieties of dogs. They're still the dog kind. We have a chocolate lab at home. Call her Abby the Wonder Dog. So how many different kinds of labs are there? We got the chocolate lab, we got the black lab, the gold lab. We got some weird neighbors. They have a meth lab. But dogs have always been dogs, yeah. Okay, cats have always been cats. The cat variety is not even close to the dog variety. I have no idea why. But we do have lots of variety there, so that also applies, I believe, to the Insectica as well. And so that's no problem. We have a lot of variation. I saw a young creation scientist right there. And then, yes, I'll get to you, Bill. Yes. Oh, why do moths look like they have hair? It's because they do. They have these hair-like appendages there. That's a good question. And what is the difference between moths and butterflies? Well, that's one difference betwixt the moths and the butterflies. Also, there's an ecological separation there. Moths are active at night, right? And butterflies are active during the day. A number of other features that are quite different betwixt the two groups. But yeah, moths look kind of hairy. Yeah, very, very unique too. Fascinating. That's why collecting butterflies is a lot of fun. A lot more fun than stamps or something. Yes, Bill.
Right, before I ask my question, I would like to make an appeal to the audience to please use the microphones.
Okay.
If you get my newsletter, you know I transcribe these meetings. And it's impossible to do it if you don't go to the microphone. I won't hear your question. So please use it.
Okay.
Here's my question. I'm curious of when you think insects were created, and were they on the ark?
Yeah, the insects were created in the days of creation there, the land animals and all, and so there's no problem there. They went on board the ark too, you know, and they took the ectoparasites with them, external parasites, which are, you know, the insects and all, and so they went on board the ark along, you know. Why Mr. Noah just didn't mash those two mosquitoes right against the bulkhead of the ark? I don't know, but yeah, so they were on the ark as well. I do want to show you this one picture. There's a beetle that water skis so fast it appears to vanish. A video clip in New Scientist magazine, way back in March of 2016, shows the water lily beetle in slow motion. A human skiing at a comparable pace would go 310 miles an hour. So that's really clipping. So I had to share that. But yeah, this is a beetle that really gets with it, you know, on the surface of the water there. So I forgot, I did want to show that. Other questions? Yes, young lady. Yeah, and if anybody else has a question there.
So before the fall of man, were mosquitoes able to suck blood, and the dragonfly nymphs that you were talking about, were they able to pinch and attack?
Yeah, so before the fall of man, you know, we would think that perhaps female mosquitoes fed on leg hemoglobin in those nitrogen-fixing plants. And that's where they got their sustenance, from the leg hemoglobin, which is a proteinaceous compound, which, and this would be a good experiment for creation scientists to see if female mosquitoes today can survive on leg hemoglobin and what? Produce ova, or the eggs. And so that would be a very fertile method of research to do. And it's in our name, Institute for Creation Research, okay? So that would be one answer there. Yeah, and then what did, and this is the predator-prey problem we have in creation science. You know, I'm not going to make any kind of apology or anything. We have a lot of unanswered questions in ICR and creation science in general, and that would, one of them would be this very sophisticated, the dragonfly nymph, and what that fed on. Maybe because of its mouth parts, it too would be able to suck on plant extracts. And plant extracts can be quite nutritious, you know, some plant extracts. So that'd be another way of doing some research on that. And I think it would be very fruitful. So that's a good question. Okay, we're drawing to a close. Ed Holyrood. Yeah. And Ed is a quintessential creation scientist. He's been around for many decades. Yes, sir.
I do not have a question, just a comment. Last summer I was in Anshan, China, and in a forest environment, and had about 1,000 gnats swarming all over my head. They're not biting or anything, just a nuisance, very bad nuisance. Then I stood in the middle of a parking lot, and about a dozen dragonflies came in, and they cleaned out that cloud of gnats very quickly. I was very thankful for the dragonflies.
And how green can you get? And it's very fascinating to watch dragonflies as they feed, because as I had mentioned in my presentation, they feed on the wing, and they actually use their 6 legs as a cage to catch these insects and then just eat them on the wing. I shouldn't share this, and I wasn't going to, but Ed, thank you, because I will share it now. Sometimes when I catch dragonflies, and there's a way of doing it, you can hypnotize them with your fingers, really, and go towards them, and they will just be watching your fingers, and before you know it, you grabbed them. And what you do is you grab it, and then, I'm sorry, I shouldn't say this, but you bend the end of their tail down up towards their legs, and once the end of their tail comes to their legs, they grab the end of their tail, they start to eat it. They don't know any better. And so I used to teach crew at Pensacola Christian College, Saturday morning crew, you know, those long crew shells with each guy has a 12-foot long oar, 8 guys, and then you got the coxswain. I'm getting some blank looks here. But anyway, and so I would, we would, since I was a teacher, they had a required nature walk, you know, before we went back to campus. And so I would do that, and it gross them out every time, but you could tell they liked it, so. Okay, so is that it then? Oh, yes, okay. Maria?
One thing you didn't say was all sciences
Oh, all science is creation science.
and all.
Truth is God's truth.
Yes,
Thank you, sweetheart.
That's, well, but on that note, I know that you guys do the discussion and stuff, but the main takeaway I always take away from your talks is a lot of people think that just because you believe the Bible, you can't believe science as well, and obviously this community is here because we don't agree with that. But clearly, what's something you would say to the evolutionary, you know, that kind of attack? One of the things that I heard from you was they don't have the beneficial mutations there, you know, and we're clearly doing science and not just spoon feeding Bible verses all day long.
Right,
So,
right. Yeah, so we're doing the research, and so Maria brought up a very good point, that all science is creation science, all truth is God's truth, and that science is a search for truth, right. John 14:6 says, Jesus saith unto him, I am the way, the truth, and the life, and that no one comes to the Father except by me. And so one thing I've learned through my years of teaching at Pensacola Christian College and then through my years at ICR, is that science and the Bible go together beautifully. They dovetail just like butter and bread, just like politics and corruption. They go together beautifully. So you've been a very good audience, and Ed, I'm going to turn it over to you.
Thank you, Frank. Very interesting presentation. I'm sure everyone was intrigued by it as I was. You said a few things there about insects and things that I really didn't know about. I used to be an avid butterfly collector, and I had collected them from all over the world, not personally, but trading, you know, and all that sort of thing. That got wiped out with the Katrina flood as well. But I got some new butterflies, and I'm going to put together another collection. Anyway, they're very interesting insects, and some of the, there is a butterfly that has about a 12-inch wingspan, and it's called a birdwing type of butterfly, and it lives in Borneo, and it is called the emperor birdwing, and it is so rare now, or endangered, that it's highly preserved, and collectors can't get to it anymore. So just a little update. Listen, don't forget about our freshmen downstairs, as always. Next month's speaker will be also a Texan from Glen Rose, Texas, Dr. Carl Barr, and he will come with an interesting presentation, as he's done before when he's been here. Not to say that Frank's presentation is less interesting, but in fact, it's different sort of thing, actually, okay? So don't miss Dr. Barr next month. And wish you guys have a marvelous weekend, and go with the Lord's blessing, and we'll see you back again. So long.